Okay, let's talk about proliferation and the cell cycle. So we need growth to produce multicellular organisms and to replace cells that are worn out during homeostasis. And there's various different parts of the cell cycle, and you can see the example cartoon of it just here. So as an overview, the main goal of the cell cycle is to produce two genetically identical cells from the one precursor. And the way this is done is through an orderly sequence of events. There are phases of the cell cycle. So the first thing the cell has to do is duplicate its contents, which involves production of replication of the DNA to produce 2 copies of the DNA, separation of the chromosomes, and also the doubling of the cell mass. So you have the same organelles will be in the two daughter cells that are produced. And then after you get the duplication of the contents, then the cell physically divides in 2. And there's 2 parts to that as well. So the important thing is for each daughter cell to get a copy of the entire genome, so you're separating the DNA. And then also each cell's receiving half of the cell contents. That's that way so that the cells don't reduce their size upon division. You don't get half the size of the cell. That happens in most cases that you get that the daughter cells are the same size as the original cell, but in some cases cells replicate with a loss of cell size. And we know a lot about the fundamentals of the cell cycle through studies of various different organisms. For instance, simple things like bacteria and yeast. And the basics of the cell cycle are fundamental and conserved across these species, but the details do vary from organism to organism. For instance, the time that it takes a cell to go through the cell cycle varies quite a bit between bacteria, yeast, and mammalian cells. So there's an important sequence of events in the cell cycle. So there's 2 major phases. Interphase consists of several phases within it, so G1, S, and G2. And there's mitosis phase, where the cell is actually dividing. How does a cell know when to divide and undergo this process of mitosis? There can be a lot of signals that act on a cell from the external surroundings that tell a cell to divide. Or in some cases, the cell can have an internal clock, and so it just regularly undergoes the process of mitosis. And also, it's a very important point which we're going to come back to, are that there are checkpoints that exist to determine whether the cell is going to continue on through the cell cycle, or if there is damage, for example, with the DNA and things are not working properly, it is not in the organism's best interests for that cell to continue to divide. It could actually become a cancerous cell, which would then threaten the life of the organism. Or it might give rise to daughter cells, which just cannot function because the cell hasn't divided properly, the DNA hasn't replicated properly, or something like that. And so in those cases where things are going badly, it's better for the cell to undergo apoptosis and die rather than continue to divide. So let's go through these phases in order. So the first one is G1 or GAP1 phase. Generally, it's the longest period of the cell cycle, but in some rapidly dividing cells, it can be very short. So in cells that have finished differentiating and are no longer going to divide, for instance, a lot of neurons, they enter what is called a G0 phase, G0 state. So they're out of the cell cycle altogether. But in the G1 phase in general, the cell is functioning normally and it's producing more organelles and proteins so that the cell is doubling in size. Basically, it is preparing for the next phase, for the synthesis of DNA. And producing these extra proteins and organelles mean that the daughter cells will be able to function the same as the parent cell. So things that regulate this G1 phase and things that regulate all the phases of the cell cycle are these particular proteins called cyclins, and their copart is cyclin-dependent kinase, or CDK. So those are the active part, and they only work when they're functioning together. So these are 2 major classes of regulatory molecules. And because they're so fundamental to understanding or to controlling the cell cycle, the discovery of these molecules resulted in the awarding of the Nobel Prize. And so there's a number of these that act in each phase of the cell cycle. And so there are some that are associated with the G1 phase, G1S, S phase, and the M phase. So in G1 phase, we have some of these cyclins that are very important, particularly the cyclin D. Well, there's several of them. The cyclin D binds to a particular type of CDK called CDK4, and then that acts through quite a complex pathway, resulting in the transcription of genes, including cyclin E. The cyclin E binds to CDK2, and that complex is then activated, which prepares the cell for S phase, moving into the G1 S phase transition. So as part of that process, then levels of transcription factors that promote the expression of cyclins and CDKs involved in S phase need to be turned on and increased at that point. So the levels of these cyclins and CDKs is going up and down throughout the cell cycle. And also, at this point, you need to inhibit or decrease the levels of molecules that inhibit entry into S phase. Assuming everything has gone correctly, the cell will then proceed into S phase. And during S phase, the DNA is replicated to produce 2 identical copies of each chromosome. And the replication, so this doesn't take forever, replication starts at many sites along the length of each chromosome. What they do is form these pre-replication complexes, which then start and replicate it. Also have to make new proteins to assemble with the new DNA to form chromosomes. Because remember that DNA just doesn't exist by itself. It's also associated with histones to form chromosomes. The chromosomes are wrapped around the histone proteins. Now, after the DNA replication has occurred, you have these 2 what are called sister chromatids, and these are held together at a point called the centromere. And one important regulatory checkpoint that occurs at this point is the DNA is checked for any incorrect copying, and whether this could result in any mutations that might stop the new cell from working. So it's a very important thing that happens here to check whether the DNA is ready to go. So the control of S phase involves S phase cyclins and CDK complexes. And one of the things these do is to phosphorylate proteins that make up that pre-replication complex on the DNA replication origins. So these are scattered all over the chromosomes, all over the genome. And the important thing that happens here is that once these bind and they start replicating the DNA, part of those proteins are then moved away from that replication site, which means that that complex cannot be activated again during that cell cycle. These things are spaced out so that part of it happens in G1, the rest of it happens in S phase, and then it moves away from it, and then that pre-replication complex cannot be formed again until after the cell cycle is completed. This means that new complexes can't be formed, and so the genome can only be replicated once, because you don't want it to be replicated twice, or random bits of DNA to be replicated 2nd or 3rd times. Just want one replication. So assuming that S phase is going correctly, the cell then moves into G2 phase, GAP2 phase. So in this time, the cell is preparing itself for mitosis. It's synthesizing the components to allow the survival and functionality of the daughter cells.